everybody, and welcome to today's private investor event with Maxwell Biosciences, which is a very exciting company that we're pleased to have our CEO and head of investor relations on. Backed by over $30 million in non-dilutive and government financing, and over 250 peer-reviewed studies from over 2 decades of R&D effort, Maxwell Biosciences develops novel patented compounds that are shown by multiple independent labs to rapidly and safely inactivate life-threatening viral, bacterial, fungal, and oncological targets. The company is a preclinical stage platform biotech company. Their initial A round of $5 million has been fully subscribed, and the board has now authorized an additional $5 million of capital that can be raised upon the same deal terms, despite the fact that the company has experienced several value inflection points. The key investment considerations are, first of all, breakthrough patented and validated technology. The company, which Josh will explain soon, has a first-in-class biomimetic drug discovery platform, which precision engineers non-peptide small molecules with 100% functional mimicry of most bioactive human peptides. The company is operating in a huge market opportunity. The peptide drug market is currently valued at $60 billion, and with Maxwell's fully synthetic compounds, they plan to see this market expand tremendously beyond. The leadership team is world-class. The co-founders, co-inventors, board chair, and senior executives of the team have deep business operations experience in this space. They've commercialized new technologies and are a proven group. They have also been involved in several IPO exits, and a deep science and biochemistry background as well. The company has a clear pathway to IPO. I know that's very attractive to a lot of our investors. They're on track for IND application of the strategic lead drug candidate next year, and they're targeting Q3 of 2023 for human trials, at which time they plan to IPO in late 2023. They've already had conversations with several American, Japanese, and German pharma companies that could produce some licensing deals and generate 9 to 10 figures worth of revenue. In short, this is a ground floor opportunity to hop on a rocket ship here. The company's seed round has been expanded, as I mentioned earlier. Minimum investment amount is $50,000. And with that, I will turn it over to the CEO, Josh McClure, and we'll do a 20-minute presentation-ish. And then we'll have 40 minutes of moderated Q&A by Mira, who's head of investor relations. And then we'll do a poll, maybe 15 minutes before the end, so that you guys can indicate your interest level. Thank you guys for joining us. Let me turn it over to Josh now. Josh, welcome.
Thanks, Jim. Good to be here. Thanks for having us. So I've got 19 years of CEO experience, and I'm excited to tell you about Maxwell Biosciences and our breakthrough infectious disease and oncology drug platform that is backed by over $30 million of non-diluted funding from the Japanese, Australian, and United States governments. A good example is DARPA's grant to create a synthetic immune system, which is essentially what we are. In addition, I'm going to be telling you about 2 major problems we're solving and how our solution can make a transformational difference in humanity. I'm going to tell you about the tremendous market size and how our technology, our team, and our business model have been strategically developed and designed to create great returns for shareholders. I also want you to know that we're in the final stages of our funding seed round, as Tian said. It's oversubscribed, but we have board approval up to a maximum total of $10 million. So the $5 million round now is expanded to $10 million, but we're going to probably cut it off before that, because we've got right around $8 million or so that's already in 2 different escrow accounts already. So it's even the funding level, not just subscriptions, is pretty high already. So we anticipate the round will close next week. That doesn't give you very much time. We've got a deep dive session that's coming up in the next couple of days. So be ready for that. You should also know that we've had many investors complete due diligence. So these are, some of them are institutional quality venture capital firms that have already gone through due diligence and brought in their science team. We've also gone through due diligence with Keiretsu, which is a large angel network you're probably familiar with. So you're going to have to move pretty fast. You'll probably need to dedicate some time to diligence to make this happen fast enough to get in. And if you can't get in, that's okay. Like we're overfunded, so don't feel bad for us if you can't get in. We're already paid up for this pitch, and so we wanted to make sure that we did it. So let's get into the details. Here's our disclaimer. Problem number one that we talked about that we're going to address is that cancers and infectious pathogens are an existential threat to mankind. We know that. Viral, bacterial, fungal, polymicrobial, biofilms, and oncological threats continue to evolve, evading drugs today and overwhelm our innate immune systems. Problem number two that we're going to talk about is that naturally occurring peptides used by your body, and when pharmaceutical companies try to make drugs out of those to fight pathogens and cancers, those peptides are unstable and quickly break down due to natural enzymes in your body and also enzymes released by pathogens as a defense mechanism against your immune system. So peptides are unpredictable in human trials and are also expensive to manufacture. So that's problem number two. Our solution is Maxwell's Clarimer branded platform technology creates synthetic biomimetic small molecules. These are non-peptide compounds, so totally synthetic, non-biologic, that can mimic and enhance almost any of the 1,000 to 3,000 peptides active in your bloodstream. And those peptides are performing a wide variety of functions, not just your immune support, but also hormonal, metal transport, copper transport, etc. So we're starting with cathelicidin peptide mimic, which is human cathelicidin antimicrobial peptide. We're mimicking just a very small portion of that peptide in a very much smaller molecule. The primary immune peptide in your body, this cathelicidin antimicrobial peptide, is used to fight cancer and infections. So these cathelicidin peptides are extremely well studied. There's literally thousands of studies. So we can actually use those studies to better understand and precision engineer our compounds. Here you can see a list of the institutional confirmed results so far. So these are independent institutions that have confirmed our results so far on our non-peptide small molecule Clarimer branded compounds against many serious and life-threatening pathogens and tumors. So our Clarimer compounds so far display 100% biomimicry of the cathelicidin antimicrobial membrane disruption mechanism of action, and have shown enhanced potency, safety, and stability. Don't take our word for it. Fauci's own team at NIAID reports our compounds as highly potent. I just got off a call with them. I was actually a few minutes late to the prep call for this with your team here, Tian and team. And they said that they will be prioritizing our in vivo testing in animal testings against coronavirus, and that we are highly recommended for moving forward into therapeutics. So that's really incredible. It's a big victory for us, and here's why. So our Clarimer compounds, as confirmed by NIAID, compare against data research by the Scripps Research Institute as among the highest potencies of 12,000 new chemical entities tested against coronavirus SARS-1, and NIAID reports our compounds are the first safe drug candidate to demonstrate pan-coronavirus efficacy with confirmation against SARS-CoV-2, MERS, which is the Middle Eastern version that was highly lethal, that was miraculously stopped in the Middle East, and also coronavirus beta. They've also confirmed pan-influenza potency. We're now talking with NIAID about testing against influenza as well in animal models. NIAID isn't alone. Many government and academic institutions around the world have confirmed our efficacy, including the Japanese CDC, Stanford University, and New York University, and the Baylor College of Medicine reported our compounds as a breakthrough drug. So something that Tian mentioned, Maxwell has leveraged over $30 million in non-dilutive awards and grants to support most of the research efforts so far. There's over 250 peer-reviewed studies validating our science that have been published based on those awards and grants, collaborations with universities and institutions all over the world. We can send you a link to our data package where you can learn more about these published study results, including virucidal potency. So not antiviral, we don't slow down the virus, we actually completely destroy it and eliminate it from the system against both RNA and DNA viruses. Bactericidal potency against 45 plus multi-drug resistant strains, including total drug resistant tuberculosis. Gold standard multi-drug resistant cancer efficacy. So these are cancer tumors that are resistant to chemotherapy, multiple types of chemotherapy, and are also not able to be targeted by modern immunotherapies. We've also demonstrated safety in these cancer studies. This is the published study here, published in the high-impact peer-reviewed journal PLOS ONE, that reported significantly inhibited tumor growth in a human breast cancer xenotransplantation model, which is the gold standard in the pharmaceutical industry. That's when they take a patient's actual tumor and transplant it into a mouse. And there are no noticeable acute adverse effects in the mouse. We also show fungicidal potency against drug-resistant deadly fungal infections. There, Dr. David Corey is the one who performed that study and is a medical doctor at the Baylor College of Medicine who works with immunocompromised cancer patients. He often watches his patients slowly die, not just from cancer, but unfortunately from opportunistic fungal infections because these cancer patients are immunocompromised and immunosuppressed. And they breathe in these mold spores, which everybody does all the time. They don't have that immune system, and so they can get deadly infections from these mold spores in their upper respiratory tract. So these types of infections are currently completely untreatable because there are no fungicides other than our own that are safe in humans. And so as I said, they're often deadly. Dr. David Corey is a world-leading expert in this field, maybe the top scientist in the world on this field, and for over a decade has tested various solutions without success. So imagine persistence, never seeing success, and then he tests our drugs, and he said, this is a breakthrough. I can't believe this is happening. He had to confirm the results again. And when he compared the remarkable effectiveness of Maxwell's Clarimer fungicides to current experimental and sometimes highly toxic antifungals in immunosuppressed mice, he called me to say, this is a breakthrough, Josh. Here is the verified data from his lab showing a significant reduction in fungal units, while his treated mice also remained happy and healthy with no signs of toxicity. So this study and many others show how our solution can address problem number one, the ongoing threat posed by infectious pathogens and cancers. Now, without getting too deep into the science, which we can save for the Q&A sessions and deep dive, let me share some important points about how our compounds work, the mechanism of action. Nature Scientific Reports published our compounds as fast killers of drug-resistant pathogens. So these are medically unmet needs by targeting a pathogen-specific marker, the same one that your immune system uses, called phosphatidylserine. I'll say that again, phosphatidylserine. So your membranes in your body are made up of phospholipids. So it's a fat layer that surrounds the cells. And phosphatidylserine is expressed on the outer leaflet, the outer side of that membrane, when your cells are infected, when they're sick, when they're cancer cells, or even old cells. It's not expressed in a healthy human cell. None. There isn't any. Otherwise, your immune system would be attacking your own cells. So this is a pathogen-specific vulnerability or cancer-specific vulnerability, and it doesn't exist in healthy human cells. That's what we're targeting. So here you go. This pathogen-specific phosphatidylserine membrane vulnerability, or what allow your immune system peptides to kill them. We use that same vulnerability in our drug discovery process, and Maxwell's technology is well published to disrupt pathogen membranes, completely inactivating the pathogen or tumor while completely ignoring healthy human cells. Again, don't take our word for it. The University of Texas Medical Branch used electron microscope imaging to confirm viral membrane disruption by Maxwell's Clarimer branded technology. Here is untreated virus under that University of Texas electron microscope, both RNA and DNA virus. So HSV is an RNA virus. SARS-CoV-2 is a DNA virus. So these very disparate viruses. And the similarity that they have is they both have membranes, which means they both express phosphatidylserine, which is that marker that we go after. You can see clear images of that sun-like corona there in the bottom image of SARS-CoV-2. In the second column here, the UT scientists captured treated virus, but with partially disrupted membranes. So this is like a bubble, like blowing a bubble like you did with like soap bubble, right, when you were a kid. And this is like a photograph of that bubble mid-pop. It's almost like an art photograph, only with an electron microscope. So partially disrupted membranes, just like bubbles, do not exist in nature for more than a microsecond, illustrating a verified mechanism of action study result. For UT's grand finale here, in the third column, you see fully disrupted membranes. So compare that disrupted splat of goo there, that used to be SARS-CoV-2, to the untreated intact coronavirus on the left. So Clarimer-branded compounds accurately mimic the cathelicidin binding and disruption of viral lipids while completely avoiding human cells. Similarly, they disrupt the viral membrane in a highly specific mechanism of action. And then you've got this layered mechanism of action that mimics the immune system. So disruption of the viral envelope, then rapid binding of the internal proteins of the virus and the nucleic acids, the RNA and the DNA. This new technology is desperately needed in the clinic, and as you can see, our compounds are highly effective at destroying these infectious pathogens and addressing problem number one, as confirmed by NIAID, and now we are officially prioritized to move forward into animal testing and animal models of this infectious pandemic. But what about problem number two? So pharma companies who are seeking new therapeutic drug candidates love peptide biologics because of their adaptable folding properties. You've probably heard of protein folding. Peptides are just small proteins, right? And so they fold and they move and they do things within the body. No other small molecule does that other than ours. So our small molecules actually fold and move. That's how they're able to do this really outstanding stuff. And so they're essentially performing the functions of life, what natural human peptides do. But pharma hates peptides because of their clinical shortcomings due to the innate instability that is caused by enzyme degradation. So your enzymes in your body, everybody has a different amount of enzymes, right? And so imagine trying to do a statistical study and everybody's different. Every patient is different. Every time you, as a doctor, you administer that drug, it works in some people, it doesn't work in others because it doesn't stick around very long because it is a biologic that your body is able to break down. Our drugs do not have that problem at all. So here you see pharma companies know, as the rapidly descending red line there shows, that peptides break down very quickly in vivo. They're designed to do that, right? So you need a drug, you need a druggable medicine in order to get through the FDA. Peptides are very unpredictable drug candidates for human trials. How do you fix that problem number two? Here you go. Our patented Clarimer small molecules are 100% biostable. We've repeated this study so many times over the past 10 years, and totally predictable in vivo, as the horizontal red line shows. That just keeps staying at 100%. The stability of a small molecule combined with the power and protein folding properties of a peptide is highly sought after by pharma. This is the holy grail of drug technologies. Preclinical studies show the Clarimer platform safely mimics the desirable pharmacological properties of peptide therapeutics while overcoming problem number two, peptide weaknesses. In particular, there's an excellent animal safety data. We've got a lot of it in our data room that you can review. You can bring scientists and doctors to that deep dive diligence. You don't have a lot of time, but you need to get them interested and looking at this so you can invest quickly. We've proven biostability over and over again in animals and in labs. And we have high potency in animal studies as well as high levels of safety, which allows for lower dosing, which is less expensive, scalable manufacturing. We've scaled manufacturing up to the kilogram. Up to 100 grams. We are now going to kilogram. In addition, our compounds have a smaller molecular size. They're small molecules, and they're engineered to precise target-specific structures like phosphatidylserine, but we can engineer them to target many other types. The broadest addressable market using just the cathelicidin mimics, so just targeting phosphatidylserine, is in the range of about $370 billion annual revenue, disrupting about a $3 trillion chronic disease market. So we focus in large areas others can't touch, serious, otherwise untreatable, hospitalized conditions that are completely unmet medical needs, where we can get breakthrough designation, we can move into emergency use authorization to rapidly prove out the platform and save a lot of lives, hopefully. So as I mentioned before, our platform leverages over $30 million in government funding. So thank you, American, Japanese, and Australian governments for funding this, because it's been a very competitive process to do that, but we keep winning. And I think that we've got a lot more money coming. Obviously, we've been in NIAID-sponsored research, and they're about to sponsor the animal trials as well. Those are very expensive, so we'll add to that number. We've got 20-plus years of research by top U.S. scientists, the U.S. Department of Energy, National Institutes of Health, Stanford, and NYU to develop. Those scientists have been rounded up into our team now. So we've rounded up that proprietary know-how on this technology and the patents. There's strong composition of matter patents, as you see here. These are granted, issued patents that we either own outright, or we've owned the core patents outright, or we have licensed patents in from collaborating universities that have collaborated in those grants and awards. So let's talk about the team. The Maxwell team has a deep bench of dedicated experience in life sciences, drug discovery, and development. Our team brings together both business execution and exit capabilities, right? So for investors, that's what you want to look for. How do you get your money back? And then we're combined with world-class academic, scientific, and drug development experts. In addition to my 19 years of CEO experience, I have a background in big data analysis, very important for clinical trials. My background is in big data computation and analysis, so I can actually write software. I can create computational databases. I've done that for this company. That's how we're able to manage so many different drug candidates, and each one has so many different potential clinical targets, right? And so we're constantly computing that data within that computational database. Mark Pomeroy, our COO, is a former Johnson & Johnson VP of R&D. Steve Peirce, who's chair of the board, is an oncology biotech innovator and has chaired multi-billion-dollar IPO exits. Both of his IPOs have set records. He's on track to potentially do that with this company as well. Dr. Annalise Barron is a Stanford University bioengineering co-inventor and scientific co-founder. She's one of my best friends. She recently won the NIH Pioneer Award, worth several $1 million for her work in this field. We've tallied some of that up into our $30 million for this company. And this is interesting for people who worry about the old boy network and about historically underfunded minorities. She is a historically underfunded founder class. One, she's female, but she's also 1/4 Quechua Native American and 1/4 Latina. So Dr. Kent Kirschenbaum is our chief scientific officer. He's on loan from his chemistry professor role, tenured professor there at New York University. So he is shepherding those NYU patents through to the clinic. Dr. Sheetal Valley is our director of drug development with 10 years of drug development expertise. Obviously, we have a lot more people than what are on this slide. in many different departments, but those are really the ones that we wanted to highlight today. Here's our drug platform pipeline. Our preferred targets are the most serious indications addressed in a hospitalized environment because they represent very large, completely unmet medical needs. So we're only targeting indications where the drugs didn't work, right? These are indications that are refractory to all current drugs, which gives us the chance to get emergency use authorization and lots of really good N of 1 prototype data, right? Where once we do our IND, initial, our investigational new drug application with the FDA, and we pass that, which is fairly easy to pass, then doctors are able to create a single patient experiment, which is called a prototype. And so they do this proof of concept with these patients because all other drugs have failed. So that's under the right to try law that was passed. So that data optimizes our chance for FDA breakthrough and emergency use authorization, and shows us that the drugs could actually potentially work. Internally, chronic rhinosinusitis, or also referred to as CRS, is our strategic lead indication, and I'll go into a little bit of detail why. I talked about SARS-CoV-2 and influenza and all these really sexy things. Cancer is a very shiny thing to go after, right? Chronic rhinosinusitis, come on, that's not very sexy. Like, everybody has that. My nephew has that. I can't stand the way he talks and snots all over me. That's why we're going after it, because 15% of the population has a highly valuable indication that is completely refractory to current drugs. I guarantee you, your nephew has already been on antibiotics for that snotty nose, right? So that is a massive indication. It's huge and completely overlooked by many pharmaceutical companies because it is polymicrobial. It is multiple different pathogens are infected then in the sinuses, fungus, bacteria, maybe some viruses, biofilms, different types of pathogens are in there. And so not a single drug will work, right? And so there's nothing for it. So our drug, as I've said, one compound, single compound, is effective against viruses, many different types of funguses, many different types of even drug-resistant bacteria. We tested against 45 plus different strains. There's only a few of the strains that have been found in chronic rhinosinusitis that we haven't tested against, and we'll get those results back in early January. But that'll be a little too late for you to invest unless somehow you can leverage your way into a $100 million round, which is what we're going to be starting in Q1 and Q2 of next year. So we already have verbal commitments for $20 million in that Series A from venture capital firms. So we're on track for an IND application in 2022, and we target Q3 2023 for human trials, as you can see here. So before the closing of the Series A, we are expecting to get revenue, that's right, prior to FDA approval. And how would we get revenue prior to FDA approval of our compounds? That is because of this right here. So we target Q3 2023 for human trials. As I said, how do we get revenue prior to that, right? And that's partnership royalties, licensing, and spin-outs. And we have a deal that I'm going to talk about right in just a second with a $16 billion livestock pharmaceutical company where we can actually get revenue prior to our next round, not just prior to IPO. We may be able to actually hit further milestone payments with that company prior to our IPO. So imagine what the valuation is going to be. This is a massive indication too. So we're working with this company on African swine fever virus, which is absolutely decimating multi-billions of dollars worth of pork in China right now. You can Google it right now if you've never heard of this before. African swine fever virus. It's creating not just decimating herds, but then a biohazard. They have to bring in chemical sprayers and bleach, and then bring in construction equipment to actually take out the soil in that whole area before they can put pigs back into it. And the USDA is absolutely petrified of this coming to the United States. It just showed up in the Dominican Republic. So USDA has just allocated $500 million to target it, and all of our data shows that we are the only company in the United States that is targeting this indication. Another not sexy, right? Nasty pigs. It's not humans, but it is a massive money opportunity for us. So again, we're already in talks, as Tien said, with several American, German, and Japanese pharmaceutical companies that could be worth 9 to 10 figures. And while we're pursuing FDA human track opportunities, we were approached by this large multi-billion-dollar animal health pharma company to address this multi-billion-dollar livestock problem that is a horrible problem, devastating in China, and is turning into, looks like it could be a food stock animal pandemic. So we've got a deal in place, as I said. That's not just hype. We actually have an inked deal in place that could be worth hundreds of millions of dollars, non-dilutive dollars, to us and our investors prior to the IPO, and we're hoping to get somewhere around $15 to $20 million prior to our Series A from that as well. That'll really blow up the Series A. So, as I said, we now have that deal in place. I'd say probably high probability for revenue prior to IPO, and I think we've got a pretty good chance of revenue even prior to the next round. Scientific results are great. Now let's do a little bit of back of the envelope calculations on what an exit opportunity might look like for your investment. So you can kind of multiply what you might invest in a company like this, a comp. So let's look at recent IPOs of platform biotech companies. Maxwell is a comparable biotech platform leading the world in this type of drug discovery. So with over 10 years and $35 million fueling previous development, targeting large untapped markets in cancer and serious infectious indications. So preclinical companies similar to ours, as you see here, have recent IPO comps with pre-money valuations upwards of $1 billion. So after IPO, we're looking to hit valuations upwards of $100 billion as the world's top drug discovery platform. That may seem unreal. Bring in your scientists and bankers and take a look at this and do an estimation. Even if 50% of what I said just now was complete bull, completely inaccurate, even if 50% of what I've said is inaccurate, we are still on track for a massive post-IPO valuation, assuming that even half of what I said is proven to not work out. So let's look at that lowest valuation, a preclinical drug platform IPO exit in 2017, prior to the white-hot biotech IPO and SPAC market we see today. Using this comp, ROI on a $50 million pre-money valuation comes out to about a 25x return. So I don't know what that means for your portfolio, but if you invest $100,000 into a company that is valuated at a $50 million pre and you get a return like that, you're looking at somewhere around $2.5 million as your paycheck in a preclinical IPO. So this is even before the IPO, before the company goes into the clinical trials. So our cap table includes many angels and biotech savvy investors and VCs. David Evans Shaw is a founder of several multi-billion-dollar biotech companies. You can see here, he's the lead investor on this round. Here are the terms. We're now raising on a price seed round, offering preferred shares at a $50 million pre. Now, we've already hit multiple milestones since we started this round at that $50 million pre. So we're definitely closing off at December 31st. Don't bother to even ask for any extensions. We're already overfunded. So if you get your money in before December 31st, then you get in. If you don't, then you don't. So we're using that money to fund major value inflection points, drug substance manufacturing, our investigational new drug application and phase 1 clinical trials, leading to licensing revenue, hopefully prior to our next round. And we continue to leverage large non-dilutive government grants and ongoing collaboration with NIAID. As I said, we just got prioritized to go into animals. And we have an animal health pharma deal in place already. We've also got a collaboration with the Japanese CDC on hepatitis B, which triggers liver cancer, which is a really big deal in China and Japan. So we're currently in a board-approved oversubscription. And then as a courtesy to our investors, we told them that we would leave the round open to December 31st, and so we're doing that. Key takeaways here that we have a disruptive platform, and that's a little bit of an understatement, I think, with multiple shots on goal, excellent animal efficacy and safety data, and an existing breakthrough NIAID collaboration in a time when NIAID has been allocated billions to fund the exact thing that we do, antiviral therapeutics and prophylactics. So that's a lot of momentum, and investors have a rare opportunity for a large biotech exit within 2 to 3 years at a potential maybe 25x return. So I'm happy to take questions now, and we can also invite you to attend our deep dive sessions next week that we have specifically arranged for your group. And our chief scientific officer will be joining me for those sessions as well. So make sure you bring medical doctors, scientists, anybody who you trust their opinion on this potential investment. So open to questions now.
Hi, everyone. Happy to take questions. I know I did see one in the chat, so maybe Joshua, I'll start with that one because obviously that 30 million of non-dilutive funding is quite impressive. I know, back summer of 2020 when I first discovered your company, that plus the great safety data plus the impressive team were key reasons why I invested. And then recently joined the team. But can you talk a little bit more about the nature of the partnerships and working with those governments and, and even big pharma companies, which you mentioned we've had some talks with?
Sure. So it's interesting when you start as a small biotech, you have about a 1% hit rate when you reach out at conferences and things like that. So it's really a matter of just how much rejection can you stomach? We've had 6 years of that. And now those 1% that have taken conversations with us, we've cultivated those relationships over a long period of time. And in the process, we found a biz dev agency. So it's sort of like an agent in Hollywood, right? Where they know all of the pharmaceutical companies, especially the individuals. So there's tens of thousands of people in some of these pharmaceutical companies. So you need to, it's a person-to-person relationship. You have to find the people that are interested in very early drug development programs where they're willing to license and pay money for very early drug candidates, which is where we're at. And so these are generally people in the mid to early stages of their business development career. They're hungry for disruption. And that's what we want, is because it's a very disruptive drug platform that could actually cannibalize some of the programs that are in that pharmaceutical company, but they would prefer to cannibalize their own program with revenue that they're taking than have somebody else do it, right? And so that's generally a younger person, mid-career person. And so that's what our agency is working on, is going after those people to get that revenue. We want to make sure that we get as much revenue as possible prior to the IPO, because it's completely unheard of to have a biotech IPO with revenue on operations, right? So not revenue from like a computational database that I programmed or something where we spin that out or something. That's not what we're talking about. We're talking about revenue on operations prior to the IPO. We're bootstrapping this thing, and it's pretty rare to have a biotech bootstrapped. It generally takes around $1 billion to do that. So that's the pharmaceutical industry right now. We happen to be in the middle of multiple pandemics. We've got a livestock pandemic, animal livestock pandemic in one of the most numerous and largest animals, right? So that will help us scale up production on our drug platform. We're using analogs of our human drug candidates for the animal studies. So going into livestock, we won't be using the same compounds that would go into the clinic for human trials. They're analogs that would be licensed only for veterinarian and livestock applications. But you might know 60% of antibiotics are used in livestock. They are 60% of total antibiotics produced in the world are used in livestock. So it's a massive area for us, and it will allow us to bring the cost of goods and our efficiencies in manufacturing much improved, right? So it'll directly impact our profitability on the human side. It'll also allow us to build our own manufacturing facility and retain the proprietary knowledge and know-how that we need to build this new drug class. It's a completely novel drug class. Nobody knows how to build it like we do. So it has been described as a sub-nano biotechnology that mimics life because it folds, right? It folds like a peptide or protein. And so how do you build something that needs to fold like that? Nobody knows how to do that. We're the ones that have pioneered the scaled manufacturing for that.
Awesome. Excellent. Well, I know Rob has a question as well, so I'm going to get him to ask it directly.
Thank you. Josh, a good presentation by you all. I was following up on this swine fever, call it challenge/opportunity. I've spent some time, and we've invested in both Africa and in Asia, and we've done some things with Ross and Purina. You might be familiar with them in terms of their agri side, et cetera. Can you speak on how the dialogue went with this group choosing you all?
Yeah.
And I think, to what extent do you have either goodwill or exclusivity for what period? Because that is certainly a tailwind going into any either raise or listing.
Yeah, we have really goodwill with them, and we're being very clever with the partnership, right? So generally on a biotech's first partnership with a large pharmaceutical company, and they're a $16 billion pharmaceutical company, you have to be willing to sort of like take the bad end of the partnership, right? Just like a new actor or a new artist or something like that. Generally, the big players get the lion's share of the deal. So what I did was I left out the specifics on how much anybody is going to get paid and focused on those points that you actually mentioned, was that they will work with us for a certain exclusive period of time. I can't go into the details, but then that they will pay for the in vitro and early development costs, not paying us any revenue until we hit a certain milestone. That milestone is dependent on a test that is to be done within the United States. It can't be done outside of the United States. It has to be done in a government licensed lab in the United States. So I happen to know that there is only one lab in the entire United States that is licensed to test against African swine fever virus, right? So we reached out to them and said, we want to come out and meet with you and educate you on our novel drug class. And so we waited a couple of weeks later and came back to them and said, hey, we've got this partnership for African swine fever virus. And we wanted to know, like, when would you be able to get that virus in stock? Are you performing any tests on it now? And they said, oh, no, we're not performing any tests on it now, but we have had a large pharmaceutical company reach out to us, and so we'll just order it at the same time. So we know that is the exact same lab. That's the exact same lab that they're using. So we're doing it as well. So they're paying for the development. We have a completely separate program that is also doing it, right? So we're paying for it as well. So we'll have access to all of the data that they're getting when we negotiate who gets paid what, right? So that's, I'm looking at it from every angle. So I hope I answered your questions, Rob. I understand. I've been a CEO for 19 years. I've done these kind of things before. I know how they work.
That's fine. Thank you.
Excellent. No, I saw one person go off mute earlier, so I'm not sure, Bae, if you had a specific question. I know I saw in the chat you're looking forward to getting the materials, the deep dive kind of data room link, the deck. So definitely I'll be following up with folks as well. So I know, Josh, in the earlier question, it talked about some of those relationships with governments. I know the Australian government is doing some interesting work and might also be engaged in some of the clinical trials. Maybe talk a little bit about kind of that relationship and those different indications.
Sure, yeah. So we will be doing an expanded phase 2 type trial for chronic rhinosinusitis in Australia. So our schedule, which I showed prior, was we will do the investigational new drug application with the FDA. Then we'll do a phase 1 safety study, which is with a tiny number of patients. It's the way it's always done. It's not statistically relevant, but they're just trying to make sure that it's going to be safe before you move into larger groups, right? So then in parallel with the phase 2, we'll be doing a phase 2 efficacy and safety study in the United States under the umbrella of the FDA supervision. And then at that very same time in Australia, which law allows us to do, we will be doing a very large statistically relevant trial in the same indication. And what we're going to do is we're going to select comorbidities associated with chronic rhinosinusitis, right? So in chronic rhinosinusitis, what you see is statistical correlation to diabetes, cancer, premature birth, old age. All of those are associated with immunosuppression and immunocompromised patients. So there's so many chronic rhinosinusitis is essentially a symptom of your immune system is not able to police your sinuses, right? So what are the other comorbidities there, right? So what we're doing is we're saying, okay, we want to select in the Australian study, we want to select for an exploratory study that has, an umbrella of chronic rhinosinusitis, but underneath it, diabetes, different types of cancers, different types of other chronic diseases. And so that way we can see, okay, we gave this compound to chronic rhinosinusitis. Now, what does the data say on each one of those? So you can see I'm a data guy. Like my former company that I sold out of that company to fund this company it was a commercial real estate big data company to evaluate commercial real estate and sell that data to large insurance companies and others, right? So it's all about the data to me. And the data will tell us which way to go. It's a completely data-driven company. Like I can look at that computational database and it tells me where to go. Right? I don't have to make gut check decisions. So that is what we're doing. Also, we've gotten a $5 million grant that is funding a collaboration with a university in Australia where they came up with this idea, by the way, they told us about it and then allowed us to patent it, is a surface-attached version of our anti-infective compounds. So they actually are attaching it to a contact lens and making the contact lens anti-infective. You probably know somebody who's gotten an eye infection from contact lenses is a big problem because antibiotics and other things are very highly toxic to the eye cells. Our compounds are not. So that's really interesting. That also gave us another idea, or this surface-attached idea. What is one of the leading causes of transmitted, sexual disease is herpes. So we started testing against herpes simplex virus. We saw we had very high potency against that. And so we issued a patent for an anti-herpes, it's a spermicidal condom that also, we also submitted a patent for a personal lubricant for that as well. So it turns out that sperm, the very tip of a sperm uses a negatively charged phosphatidylserine in order to penetrate the egg. And so our compounds are also spermicidal. Very interesting development there. So lots of technologies to license to pharmaceutical companies. Some of you might be thinking, okay, if I have chronic rhinosinusitis and I take this compound, then it might be spermicidal. Well, I mean, if you have sperm in your nose, then yes. So we are testing, doing PK, PD studies to show that we're able to target specific tissues like the epithelial mucosal tissues in the lung and the upper airway, and that we don't have off-target effects.
There's so much, Joshua. I hate to interrupt you. There were a flurry of questions that came in while you were answering that last one. And I think it's so amazing that there are so many different indications. And just as a question around the patents, because I know that there's a great history around the patents, because someone asked, if governments have, kind of been responsible for some of the patents, do we have any obligations to them? So just speak quickly to the nature.
The core patents are 100% owned by our company. Other patents we are in licensing from other governments and from institutions. We have a science development kit. It's a SDK. In the software industry, they call it a software development kit. We're the only pharmaceutical biotech company in the world that is using that highly effective competition style technique, where we actually publish a science development kit and encourage scientists who cannot afford to prosecute their own patents to work with us. So we actually send them the protocols, we send them the compounds. All they have to do is sign a legal agreement that says that we own the intellectual property and that they get 10% of ownership in that intellectual property. And so we're in the business of marketing intellectual property. That's how we make money. And so when somebody comes in and says, hey, I just made this massive discovery, and they get 10%, we get 90%, that's like an agent in Hollywood getting 90% of the pay for an actor. We're in a really strong position there. So
we don't have conditions ourselves on any of this.
And we get to manufacture it, right? Yeah. So we're getting the revenue from manufacturing and then taking that out of the pie too. So it's almost like a record label. So it's.
I just want to jump in real sec. Sorry to interrupt, Josh. We're going to launch the poll as we're getting close to end of time. So anyone who's interested, very interested. You might have some investors, but you're not interested, or you're not interested. If you could respond, that would be great. As Mira said, you will have access to the data room, which has all of the necessary documents, the subscription documents, term sheets, and whatnot. And we will continue. Josh, you can continue. We just want to keep the poll up for another 30 seconds or so. Mm-hmm. Thank you guys for participating.
Yeah. And Kevin asked a great question saying, am I understanding that this is more like an antibiotic versus a vaccine? Meaning it doesn't stop a virus from attacking. What it does is it kills the virus itself in place. So I think he's got the understanding right.
That's right. This is sort of like antibiotics for viruses and fungus, and then is also bactericidal, and we've tested for resistance in bacteria. We show no ability for the bacteria to develop resistance. It completely gums up the bacteria. It doesn't give it a chance to secrete the gene that it uses to inform the other bacteria. So remember, genes are nucleic acids. We bind nucleic acids. So even if they were able to secrete it, our drugs would bind that gene and make it so that the other bacteria are not able to get it. That's exactly the same mechanism of action your own innate immune system uses to, otherwise bacteria would form resistance to your own immune system.
And Joshua, the question, Kevin asked another question. I know someone asked about board compensation.
2 minutes. I've got another investor.
So you mentioned Q3 for human trials, and he was asking whether that was the first date or was a prior date ever previously scheduled, and was that ever pushed back? I think this is the first time
we've never had any delays on the human trials program so far. Now we're just now starting to engage with the FDA, but again, the strategy here is to target completely unmet medical needs. There's the GAIN Act. You can look that up, which prioritizes this type of drug, where you're going after a completely unmet infectious disease. Obviously, there's reasons why the government would want to prioritize that, right? So it's completely unmet. So there's no options for the patients, right? These patients have families, and those people vote, right? And so that goes up to Congress, and Congress goes, okay, we need a law that says people have the right to try and get a new medication for something where there's no drug and they're going to die. Well, it kind of doesn't matter if the drug is slightly toxic, right? And so that's where that act came from. It turns out we have incredible safety data. We have achieved a lot of that safety data since we started the seed round, right? So you're still getting the valuation of the company prior to those milestones, which is one of the reasons why I kind of don't care if you invest or not. So
we appreciate any investors that come in. It's definitely a great opportunity. I know as someone who's invested in the earlier rounds, I know people are scrambling to get in. We are closing on a rolling close, first come, first serve. So for those who want to participate, definitely a fantastic opportunity that will be closing soon. So while I know Joshua has to jet for another meeting with several other investors who attended an event just a couple of days ago, we certainly do have a little bit of room left. We're kind of brushing up against the edge of our total commitment, but we are taking funds on a first come, first serve basis. So I continue to follow up with folks, and so as those funds come in, that will be how we accept them. So he did answer questions about safety, efficacy. Again, the animal studies have been tremendous. We actually people asked what you think are some challenges. And the human trials are where we actually have a lot of confidence because our biomimicry is on the human antimicrobial peptide. Our biggest concern was whether it will work in mice or other animals. The fact that it is working effectively there, we're very confident it will also work in human trials. That is to come. And that is where the IND and the phase ones will start into next year. So I did see a question on board composition. I know we're at the end of our time, so we'll probably have to close this off. But we're actually just have a very exciting new addition to the board that was part of a very successful public company that brings a lot of insight and experience that will be starting with us in the new year. So more on that to come, as I can't release the name of that person yet, but it is a high-profile person that you might even know or certainly know of the company. Aside from that, we have our board chair, Steve Peirce, and Joshua and Annalise, our scientific co-founder, and of course, some of those new board members coming on in the new year. So it's an excellent time for the company to be bringing in those perspectives. We also have a very strong scientific advisory board that's also been working with us for years to continue to lead the science. Someone asked when we can expect to see links to be sent. So I think Skylar will be sending a communication to folks. And as soon as I get the list of people who are interested in learning more, I'll also be following up as she copies me on those communications and forwarding in the links for our investor meeting, a deep dive that are happening Monday at 4:30 Eastern Time and Tuesday at 4 o'clock Eastern Time. 1 hour sessions, literally dedicated for the people here. We haven't invited anyone else to that session, even though we might have a few people that, if you don't mind, we might hand invite that just connected with us and might like to be part of the conversation, if that's okay. All right.
Terrific. Well, with that, thank you so much, Mira, and also thanks to Josh. As Mira said, thank you guys for joining us. We will send out immediately the links as well as the link to this recording as well as the deck. Thank you guys for your interest. This is an exciting opportunity. It is on a tight timetable, but it's it's an overly subscribed situation, and we're grateful that you guys, your board has approved the expansion of the.
Yeah, and I'm glad we had this set because I know last Thursday when we kind of set this up, since then we've had $1 million come in like wired funds. So we are very much kind of into this ending stages, and it's great that we could fit this in before the end of the year. So I'm glad that all of you could be here. And as a fellow investor, welcome you to, again, ask questions and be part of that deep dive. We'll look forward to seeing you there with our Chief Scientific Officer, who is a tremendous communicator, someone who can take the science and communicate it in a way that is understandable, and you walk away feeling like you've also learned something. I attended 3 deep dive sessions before I invested. We might not have time for that, but definitely come if you can.
Great. Thank you guys again, and happy holidays to you.